Hello, my name is Fergal O'Farrell and I would like to welcome you to Lecture 1 of the MOL213 Developmental Genetics course. As I've said before in the introduction, the book that the lectures will be based on is Principles of Development by Wolpert. If you haven't seen the orientation meeting, please check that out. Okay, let's start, shall we? Today's lecture will talk us through several points. First, identify the central questions in developmental biology. We will become familiar with the core processes of development and the terms used to describe them. This is important when it comes to being able to describe processes and say what you see in a way. We will try to appreciate the historical conceptual advances that have been made in developmental biology, sort of the change in ideas throughout, in fact, a couple of thousand years. But obviously we will focus a little bit more on the most recent changes. But it's good to know how the field has advanced over time. And importantly here, where possible, what practical approaches they used to advance theory, to change people's minds on how development occurred. And model systems, of course, have been of utmost importance in this. And we will discuss which model systems have been used historically, which are the primary ones that are being used today, and we will evaluate the pros and cons of these model systems. We will have a brief introduction to common central processes that are involved in development, particularly in early embryonic development. And then the cell cycle is, of course, important in developmental biology, so we will have a short refresher on the cell cycle. And we will discuss some genetics with some example of genetic crossing schemes that you've probably met before. So we will go briefly through that to refresh your memory. And we will discuss how genetic screens can be designed and the different types of mutations that can occur naturally or we can generate through screening or directed mutagenesis. So let's have a look at the basic stages of development in animals. We can start here with stage 1, fertilization. And that is, of course, where the sperm meets the egg. But if we have a look at the egg, we know it's a single cell, but it already has a polarity, a difference from the top to the bottom. And the top part is referred to as the animal pole, and the bottom part is referred to as the vegetal pole. And this can be important in setting up the polarity of the animal. And we'll discuss that in further detail. This is just an introduction. So after fertilization, cleavages can begin in the egg. And cleavages are a certain type of cell division where the cell doesn't grow in between. So they're called reductive cell divisions because following each division, the resulting cells are smaller than they were, right? So it cuts once here, we have 2 cells half the size of the egg. It cuts again in another plane, we have 4 cells, again 1/4 the size of the egg, and so on and so on. And cleavage continues for several stages until we reach a stage called the blastula stage. And this is basically a pile of cells in a round ball, but the ball becomes hollow and surrounds the blastocyst. So here are some of the important terms I should mention. The blastocyst is the liquid inside the ball of cells. And we can see that the blastula, again, the animal and vegetable pole look different. The animal pole is filled with cells that seem smaller, and the vegetal pole is filled with larger, yolkier cells. Following the formation of the blastula, we start to get movements, and movements in the embryo are referred to as gastrulation. So gastrulation is the next step, and the fertilized egg is then referred to as the gastrula. So gastrulation is movements. So we have all these cells on the outside, but now we get the formation of the blastopore, which is an involution, a folding inwards of the cells, pushed in at the blastopore. And the blastopore is an important site for signaling. It has an organizing function. So as cells start pushing internally into the egg, which is hollow, but we don't see it here, but we see the remainder of the hollowness here with the blastocyst, the liquid. As they push in, all these cells pushing in can pass this area around the blastopore and receive signals from it as they pass by. So when we had this round ball of cells, signaling from one cell to a cell down here, for example, would be very difficult without going through all the other cells. But now we will have movement past a certain point where, as the cells pass, they can receive signals at the blastopore. Gastrulation continues, and we will get the inward movement of tissues, which generates the germ layers. The ectoderm, typically on the outside, and the mesoderm, this middle layer, mesoderm, pushing inwards, and the endoderm, the intestines and some of the organs on the inside, right? They push in through. This is a general concept. It doesn't hold for all the systems we will look at, but in the case of this frog, this is the way it's working. So after gastrulation, the development of the nervous system can start, and this phase is known as neurulation. And this is indicated here, and they peel back part of the embryo to show us the development of the notochord, which is a structure very important for the development of the central nervous system, the brain and the spine. So the brain and the spine start developing at this stage, and the neural tissue generally comes from the ectoderm, the germ layer, the ectoderm. Okay. After neurulation, we get organogenesis, the formation of organs, and here it's important with the somites. The formation of somites, which will give rise to skeleton and muscles, and the neural tube forms here and gives rise to the central nervous system. And these are the basic stages of development: fertilization, cleavage, gastrulation, neurulation, organogenesis. But you will notice that this figure contains another stage, metamorphosis. And metamorphosis is common to many animals, insects, amphibians, fish, crustaceans, to name but a few. But importantly, mammals do not undergo metamorphosis. What is metamorphosis? As the picture indicates, it's a dramatic change in the shape of the animal as it goes from one juvenile stage to the adult stage. And this is driven by cell differentiation and cell growth. So what are some of the fundamental questions that have driven the field of developmental biology? I guess the largest and most obvious one is how does an adult come about? How are they formed? But we can break that question down into several sub-questions from what we know now. So some of these questions include how are different cell types generated from a fertilized egg? So we know that when the egg is fertilized, it's a single cell, and yet it gives rise to complex form of a human embryo and eventually an adult. And this adult will be composed of more than 200 cell types. So what process is important here? And we see that the development of body pattern, the egg, very simple round structure, gives rise to this complex, multifaceted structure or body, which has an obvious body pattern that's not evident from the starting material, the egg, and develops numerous types of organs. So how do we get the formation of these organized structures? This leads to a sub-question of What controls the organization of cells that generate these complex structures? For example, organs, limbs, the brain, eyes, very complex structures. What is controlling the organization of those cells? How are the organizing principles that will give rise to a full adult imprinted in the egg? How does the egg know what to do? And then, in order to know how useful model systems can be, experimental model systems to study development in higher organisms like humans, we need to know how extensive is the conservation of control mechanisms in these different animals, right? To know, are they useful for us? Are the same regulatory mechanisms going on in simpler animals as they are in more complex organisms like humans? And then by knowing this, perhaps we can define if there are general developmental control mechanisms. Okay, so let's talk about the establishment of body axes. So axes is the plural for axis, a single axis. And an axis is an imaginary line that divides something in half, very typically with a mirror image on the other side, for example, or around which an object can rotate. And there are several of these axes in the human body. So if we have a look at it here, we see that the human body has an anterior to posterior axis, represented by this line here, which divides the front of the animal from the back of the animal, or behind of the animal, anterior to posterior. And the human body has a dorsal and ventral, with dorsal being the back area, region where we would have the spinal column, and ventral being the region where we have our stomach or gut in the animal. And these axes are actually set up in the early embryo. And the early embryo just consists of a ball of cells, and yet it is able to set up the dorsal ventral axis and the anterior posterior axis. And later will come a left and right axis, which of course the human body has a left and right axis as well. And many animals have bilateral symmetry in that they are round in shape. They still have dorsal ventral, anterior posterior, but it's not always easy to tell if they have a left and right axis. In the human, it is. Not only do we have mirror image left and right hands, but the organization of organs inside in the body can have a left-right orientation. For example, your heart will be on slightly to your left of center in the majority of people. And like I said, these axes are set up early in the embryo, and in fact, the material to set this up is deposited into the egg by the mother. So before it's even a fertilized cell, there are maternal factors placed into the egg by the mother. And maternal factors, the most typical ones are mRNA and proteins. And the mRNA itself will generate more proteins. So it's indirectly proteins deposited into the egg that will help set up the axes early on. And in some cases, these maternal factors can be localized to specific areas of the egg, and we will discuss this. Typically, after the establishment of these axes in the developing embryo, we have the establishment of the germ layers. And I mentioned these briefly before. We have three germ layers: the ectoderm, the mesoderm, and the endoderm. The developing embryo becomes subdivided into these three germ layers during the blastula stage. They may not be visible at that time, but they are defined as such there. They become visible um, with the use of microscopes then during gastrulation, the phases where the cells in the egg start moving. Remember I mentioned this, there's an involution around the blastopore, and the cells collectively move together through the blastopore towards the inside of the egg and become determined to whichever germ layer. So also during the gastrulation or transition from blastula to gastrulation, where things start moving, the embryo becomes subdivided into major regions along the anterior-posterior axis. We see here an example of an egg which is slightly elongated to help illustrate this. We have the anterior here, posterior here, and the embryo becomes subdivided into regions. For example, the head, the thorax, the abdomen, and the tail. And this subdivision occurs through regional restrictions of gene expression, which can be initiated by the maternal factors deposited in the egg. So the maternal factors can act on genes such that only certain regions of the animal will switch on those genes. So the head will switch on a gene, but the tail won't. And they can also be graded. You have a maternal factor that is deposited at one end of the embryo. Say it's an mRNA and it starts producing protein. The protein starts accumulating at this end, but it's much less as you move towards the tail. And this graded expression of the gene product, the protein, will lead to activation of genes in a graded manner across the embryo. And we will discuss all this in more detail further on. So that was just a brief introduction to some of the processes that we're going to be looking at. And now let's have a look at some historical perspective on the field of developmental biology for a few slides, and consider what approaches were used to advance the understanding of developmental biology. The philosopher Aristotle, around 300 years before Christ, contemplated this problem of how do you develop an adult from an egg? How is a baby generated and gives rise to an adult? And he came up with two hypotheses that could be possible. That the embryo was progressively forming new structures was one of them, and this was called epigenesis, so the generation of new structures. Or was it possible that there are preformed structures, that the egg is preformed and that these simply grow bigger? And Aristotle favored the model of epigenesis. He discussed two possibilities, and he favored the model of epigenesis that we now know to be the case today, that the embryo progressively forms new structures. But it's perhaps surprising to know that up until the 17th century, preformation was the favored theory behind the development of an adult animal. So they were created. Even some people believed that the sperm held a tiny person inside it. When they looked with the best possible techniques they had, the microscopes, they thought they could even see a person inside a sperm, and that this would then go into the egg where it would receive nutrients to grow. And in fact, it wasn't until around 1840 that a new theory, a cell theory, was postulated by Schleiden and Schwann, that the egg was a specialized cell, and that it, through cell division, gave rise to the embryo and further the adult structure. So they really favored the epigenesis model and could show that cells were reproducing, dividing, and growing. And then later, August Weismann in 1880 came with the theory that an organism consists of two distinct cell types. There was somatic in your body and germ cells. The egg and the sperm were the germ cells, and that it was through the germ cells that information was passed on to the next generation. So heredity came through the germ cells. So up until then, it was speculated that insults, for example, damage to the body, could be passed on to the children. But Weizmann could show that it took insults or mutations, we could say, to the germ cells to have an effect on hereditary lines. And this is demonstrated here in this schematic, that the zygote generates the germ cell and also then the somatic cells that give rise to the adult structures. And the germ cells are preserved. And he then argued that mutations to the somatic cells that don't affect the germline would not have a hereditary effect. However, if you did have a mutation in a germ cell, this mutation would affect the somatic cells and be passed on, furthermore, be passed on to the next generation. And the germ cells could be studied further. They could study the fertilization of eggs. In particular, sea urchin eggs were important here. And they could show that with microscopes, they could observe that fertilization of the egg by one sperm, each of these contributed a nuclei. So we had two nuclei that were brought together and then fused in the new cell in the egg. And this was the act of fertilization that was required for initiation of the development of the egg into an embryo. So we got a zygote, a single cell, carrying a nucleus with contributions from both parents. And furthermore, they could then say that this cell nucleus contained the physical basis of heredity. So we contain, or the egg, fertilized egg, contained hereditary elements from both parents, and that was in the cell nucleus. And it wasn't until 1890 that chromosomes were discovered and thought to be the element of hereditary nature that would be passed from generation to generation. And it was figured out that there was differing amounts of chromosomes in the zygote, so from one of the parent cells, and which would be diploid, having two copies of the chromosomes, and the germ cells of that parent, which were only haploid. So then they could figure out that half of the chromosomes came from one parent and half came from the other. And this was in 1900. And this, together with Mendel's laws, so he had performed lots of experiments that he published in 1865 with pea plants, studying hereditary characteristics, but these were not understood until they could observe that, okay, half of the genetically heritable information comes from one parent and half comes from the other parent. So they could put this information together around 1900 to figure out how hereditary information was carried from parents to children. And then they were able to show that the consistency of chromosome numbers was maintained by a special reductive division, meiosis. So not mitosis that we have in the somatic cells, but that there would be a round of meiosis to generate the germ cells. And meiosis is reductive. It would give rise to half the number of the chromosomes. And this is then essential for the combination of chromosomes from both parents to give rise to a full complement of chromosomes in the offspring. And this is summarized here. within the somatic cells, we have a mitotic cell division where the chromosomes align, they duplicate, and then they align at the metaphase plate, and we have a round of cell division which pulls a copy of the chromosomal complement to each cell so that the cells remain 2n diploid. Whereas in meiotic cell division, we have similar first steps with the chromosomes finding each other, a round of DNA replication, giving a sort of 4n cell. And this differs here at this stage because the, the, the chromatids can find each other and from the different parental DNA, and there's a chance for exchange of genetic information via homologous recombination. But this wasn't observed by Mendel. This was only found later on, that you would not always get sort of true breeding between plants. There could be differences there. Anyway, apart from that, we then end up with a cell that undergoes a fairly normal division afterwards, except that more or less only pulls the mother's chromosomes to one side and the father's chromosomes to the other. It has had this chance for genetic exchange already, but the father cells are inherited by one cell and the mother's by another. And then we have another round of reductive cell division where the chromosome complement is split up and we are left with haploid cells, the gametes. And then, of course, we get one of these from the mother, one of these from the father, that meet in the egg and the sperm, and we restore the complement of chromosomes to 2n again at that stage, and development starts. While talking about cell division, mitosis and meiosis, it's a good chance for us to refresh on the eukaryotic cell cycle. So the cell cycle, as shown here, is an ordered sequence of events in which a eukaryotic cell duplicates its chromosomes and divides into two, and it takes approximately 20 hours. But that will vary in different animals and at different times during development. The cell cycle normally consists of 4 phases: the S phase for synthesis of DNA, the M phase for mitosis, where we have nuclear division, mitosis, and then cell division, cytokinesis, when the cells separate. We have 2 gap phases called G1 and G2 between S and M, and these phases are to allow more time for cell growth, but they also monitor the environment and check the DNA integrity and other internal processes. The M phase, mitosis, is pretty short, about an hour, and it includes several visible processes, which we'll just remind you of now, that cells exit interphase. They go into prophase, where the DNA starts condensing. We get a prometaphase, where the chromosomes are visible, and then a metaphase, where they align at the metaphase plate at opposite ends of the spindles in the center of the cell. And then the cell cycle enters into anaphase, where the chromosomes are pulled apart by the spindles at either end of the cell, and telophase starts, where the cell actually starts into cytokinesis and the membrane invaginates towards the center of the spindles. Chromosomes are at opposite ends of the cell, and the cell divides cytokinesis. And then the DNA starts to expand again, and the cell enters into interphase. And interphase is thought to be the S, G1, and G2 phases generally, which do not display clearly visible changes. So if we look at the time frame for these things, these different phases of the cell cycle, We see that in human cells, cell cycle takes approximately 24 hours to complete, where they spend 9 hours in the G1 phase, the growth phase, 10 hours in the DNA replication S phase, about 4.5 hours in the G2 checkpoint or growth phase, and then mitosis can take about 30 minutes. This is typically what's observed in cell cultures. It probably differs in that cell in the human body, but not by much maybe. So the cell cycle of, for comparison, of a rapidly growing organism, for example, yeast, which is a eukaryote, but it's a single cell, it only takes about 90 minutes to go through the whole thing and give rise to the next generation of yeast cells. The early embryos of species with large eggs, for example, amphibians and fish, they undergo very rapid cleavage divisions. It only takes about 30 minutes. This is shown here, approximately how cleavage is. And we mentioned it before, it's this reductive divisions where each cell is progressively smaller, half the size. So you don't have growth phases in between. So you have a direct alternation between synthesis and M phases without any of the G1, G2 phases. So they go really rapidly. So there's a high rate of DNA replication, and together with the constantly entering into mitosis, this precludes transcription. So because the cells are dividing so fast, they will not have time for transcription. So these cleavages depend on large reserves of RNA and protein and other things like membranes, for example, that have to be put into the egg already, right? There will be no growth in the first rounds of cell division. There will be no growth in that very early staged embryo. And we have very fast cleavages in zebrafish. Approximately every 20 minutes it takes to give rise to new cells we see forming here on top of the yolk. So people had accepted that the embryo was created by cell divisions. This begged the next question, building on this observation, was how are the different types of cells generated that give rise to the fully formed adult with, for example, these 200 different cell types and very different body composition in different parts of the body. And August Weismann postulated on the basis of discoveries of both the haploid diploid cells and the hereditary nature of the nucleus, he came with an idea that there were nuclear determinants, so factors, unknown factors in the nucleus that were important for the specification of different cell types or the determination of different cell types. And that these, together with specialized kind of cell division, asymmetric cell divisions, so cell divisions that distribute these nuclear determinants unequally, could generate different cell specificities. So in the example here, we have 4 colored nuclear determinants. The cell undergoes cleavage, and each cell inherits just one of, in this simplistic model, either inherits one or none of each of these determinants. And after another round of cleavage, each cell has only one. And in this way, you can generate distinct cell types by inheritance of these nuclear determinants from the nucleus of the mother cell by asymmetric cell division. And this model gives rise to then the 4 cell stage here, which is mosaic in cells. So mosaic implying that each cell is different. It's a mosaic of cell types already. And this model of early development is known as mosaic development. And there was some early experimentation done to test the feasibility of these models. And this was done with xenopus or frog eggs. And then what they saw was that if they had a fertilized egg, they let it divide with the first cleavage, and then they killed one cell with a hot needle. They saw that development progressed as normal for the untouched cell. With the remains of the dead cell beside it, we got the development of the blastocyst, and even to the neurola stage, we had a half-formed embryo with still the remains of this dead cell. So half of the embryo didn't form because half the cells were missing, but they would argue that uh, the determinants for that part of the body were lost when that cell was killed. So they would argue that cell fates seem to be determined at each cleavage, and that these results were consistent with the mosaic model. This was followed up by another scientist, Drisch, around 1900, choosing a different developmental model, a sea urchin larva. And they did a very similar experiment. So normally you would have a set of cleavages shown here from the two-cell stage, the blastula giving rise to the larva. And they did the same experiment, or almost the same experiment as in the frog. They removed one of these cells, which usually died. So they split up the first, after the first cleavage, they split the cells and they removed one of the cells and followed its progression. And what they saw was they developed more or less normally, giving a smaller blastula and a smaller than normal larva, approximately half the size. So this observation shoots a hole in the theory of mosaicism, that there are cell determinants that are asymmetrically distributed, and if you kill one cell, the embryo cannot form fully because you're missing half of the determinants already. And this then gave rise to a new theory of regulation, that there was a regulated development, that the cells that were left over could sense the absence of another or induce neighbor cells to take that role, that the embryo as it developed was adaptable, that things weren't determinant right from the mother's distribution of maternal factors or these nuclear determinants, that there was the embryo was able to compensate or regulate for the absence of a cell. So this gave rise to two theories, regulation and mosaic. And experiments had been done to show the existence of both. So people started to argue, well, there are experimental differences between these two approaches, right? The dead cell in the frog embryo, perhaps that interfered with development, while in the case of the sea urchin eggs, cells were separated and therefore did not influence each other. So along came a third scientist, Thomas Hunt Morgan, who was awarded the Nobel Prize, but not for this study, but for another study. He redid the frog embryo experiment, but this time not just killed the cell, he removed the cell, and he saw that the result was the same. He got a frog with half an embryo developed. So arguing that indeed there was mosaicism, but we could not argue against regulation in the case of the sea urchin egg. So then the conclusion is that there are inherent differences between the species, right? So eggs from distantly related species have a quite different composition. What we know now is that embryonic development of various animal species show different degrees of mosaicism and regulatory capacity. So you can have both this regulated development where cells communicate with each other and maybe induce a cell to become a certain cell fate. But you can also have this mosaicism where you have nuclear determinants that are asymmetrically distributed. This is known to occur in the development of, for example, the nervous system as well in many organisms. Both cases of asymmetric cell division, where you distribute determinants, and then those cells actually afterwards, they talk to each other in a regulatory fashion. So both these models can be correct for the different animals. And what's really the case is that there's a different contribution of these two ways of generating different cells in different species. So these experiments in the sea urchin that revealed this mode of regulatory development led to the discovery of the process of induction and how important cell interactions could be. So when we removed half the sea urchin cells from the egg, the leftover cell in its rounds of division could induce neighbor cells. One cell could induce a neighbor cell towards a certain differentiation to become a certain type of cell to fill in for the absence of that cell that was missing. So this is induction, a process whereby one cell or cell group affects the development of another group of cells nearby through signaling. Grafting as an experiment was very important here. So when I say grafting, I mean that they would cut one piece of the embryo out and put it into the new embryo and see would it become part of the adult and would that adult look like normal, or would it the developed adult have a phenotype? Would it look different from the normal adult? They used related species of salamander that had different pigment in the cells so that they could see under the microscope. And then the idea would be if you took a piece from here and you put it here, how did the adult look? And because it was coming from the same region, that it would have the same cell fate, the adult would look normally. But they found that when they took an area of cells from around the blastopore and put that in a different part of the animal, the other embryo, that this region had organizing function. So it was able to induce cells to become something different from what they would normally become in the adult. And what they saw actually was that they could induce a second embryo on top of the other developing embryo. This is shown in a cross section here. You have the expected embryo, but if you put in this part of the blastopore from another animal grafted into another region on the embryo, you would get secondary structures that are induced. So from both, with contributions from both the original tissue shown in yellow here, but also it would induce tissue that would normally give rise, say, to the ectoderm, instead now taking part in forming a new secondary notochord or neural tube, right? And then by taking all these different parts, they found that this, and doing the experiment, they found that this part around the blastopore was very important. And this became known as having an organizer function, that it could organize surrounding tissues, or it became known as the organizer.